So, hello and welcome back, all dear students. The topic of today's talk is about the topoisomerase type 1 activity. That how does this enzyme act and what is the mechanism of this enzyme action. आज की इस क्लास में हम लोग बात करने वाले हैं टाइप 1 टोपोआइसोमरेज और उसके मैकेनिज्म ऑफ एक्शन के बारे में कि टाइप 1 टोपोआइसोमरेज किस प्रकार से वर्क करता है. So type 1 topoisomerase is an enzyme that I have already told you is involved in introducing a twist in the DNA molecule. कोई भी एक DNA molecule है उसके अंदर ट्विस्ट लाने वाला जो एंजाइम है वो है टाइप 1 topoisomerase. Type 1 topoisomerase has the ability to introduce a twist within the particular DNA molecule, and it has the potential to relax a negatively supercoiled DNA molecule by increasing its linking number until it becomes relaxed, जब तक कि वो relax नहीं हो जाता. So let us look at the type 1 topoisomerase activity.
The topological state of DNA affects its functioning in the cell. Cells use the torsional strain of negative supercoiling to promote strand separation for processes such as DNA replication and RNA transcription.
So, negative supercoiling of the cell is being used in several processes. Cell के अंदर negative supercoiling. What do you mean by negative supercoiling? Negative supercoiling is a type of supercoiling when the DNA is much more relaxed. When the DNA is much more relaxed, then the DNA can be effectively opened. If the DNA can be effectively opened, then this negative supercoiling can be used in the cell in order to promote the process of transcription to take place. Negative supercoiling जो भी सेल की होती है वो इस बात को इंश्योर करता है कि सेल का ट्रांसक्रिप्शन इफेक्टिवली हो ताकि ज्यादा अमाउंट ऑफ आरएनए बनाया जा सके, सो दैट लार्ज अमाउंट ऑफ आरएनए कैन बी प्रोड्यूस्ड बाय द सेल. नेगेटिव सुपरकॉइलिंग जैसे कि आप लोगों को बताया गया है एक एबिलिटी है जिसमें सेल अपने डीएनए को ओपन कर सकता है. नेगेटिव सुपरकॉइलिंग इंश्योर्स द ओपनिंग ऑफ द डीएनए बाय द सेल. You already know that the human genomic DNA has a superhelical density of minus 0.06. That is, our DNA is 6% negatively supercoiled. हमारा DNA 6% negatively supercoiled होता है. तो ये हमारे DNA की एक प्रॉपर्टी है. Next.
However, if DNA becomes too negatively supercoiled, the strands will separate too often, making the DNA more susceptible to degradation.
तो देखिए अगर एक DNA ज्यादा negatively supercoiled है, then what is going to happen? If the DNA is much more negatively supercoiled, then that DNA will be effectively lost. क्योंकि वो DNA बहुत जल्दी ही open हो जाएगा और अगर कोई DNA बहुत जल्दी open हो जाता है, then the stability of the DNA is being lost. If the stability of the DNA is being lost, then the DNA will not be able to maintain itself. DNA अपने आप को maintain नहीं कर पाएगा. DNA के जो strands हैं वो एक दूसरे के साथ में separate हो जाएंगे. So negative supercoiling, if it is too large, it will cause the DNA strands to be separated from each other.
The topological state of DNA is controlled by a class of enzymes called topoisomerases.
So this is most
Topoisomerases
important.
are divided into two broad classes, type 1 topoisomerases and type 2 topoisomerases.
So these are both the classes of the topoisomerase have the ability to transform the DNA from one topological model into another. दोनों ही तरीके के टाइप 1 और टाइप 2 टोपोआइसोमरेज के पास में एबिलिटी है कि ये ट्रांसफॉर्म कर सकते हैं एक तरीके के डीएनए को दूसरे तरीके में. ठीक है, दैट मीन्स दे कैन चेंज द सुपर हेलिकल फॉर्म ऑफ द डीएनए मॉलिक्यूल. ओके, नाउ we have already seen that how these molecules can alter the topological state. Now what we have to understand is the mechanism of action that these molecules or the enzymes involves in this particular reaction to take place according. Now moving forward to the next slide.
When you have completed this exercise, you should understand how many type 1 topoisomerases can increase the linking number of a DNA molecule by cleaving one strand of the DNA, passing the other strand through the nick, and ligating the nicked strand.
Okay, so the type 1 topoisomerases are a type of topoisomerases that have the ability in order to resolve the supercoiling present in the DNA. They have the ability to make a nick over one strand and pass the other strand out of it so that the resolution of the two strand can be made. एक strand को क्या करेंगे, एक strand को कट लगाएंगे, उस strand को दूसरे strand में से पास करा देंगे so that the resolution of the supercoiling can be made. We have to understand how passing the other strand through the nick and ligating the nicked strand changes the linking number. हमें ये समझना होगा कि किस तरीके से ये linking number को हम change कर सकते हैं एक form से दूसरे form में.
Note, this animation provides an example of the reaction pathway used by many, but not all, type 1 topoisomerases. These type 1 topoisomerases can only bind to segments of DNA in which the two strands are melted, that is, segments where the strands have separated.
Okay, so here we have to understand that this band represent a double helical strand of the DNA, and these are the two strands of the DNA which have been separated or melted away from each other. तो ये जो band, ये patch आपको दिख रहा है, ये brown color का, ये क्या है? ये है एक डबल स्ट्रैंडेड डीएनए और यहां पर है सिंगल स्ट्रैंडेड डीएनए जो कि बन गया है डबल स्ट्रैंडेड डीएनए के मेल्टिंग से, डबल स्ट्रैंडेड डीएनए के सेपरेशन से. नेक्स्ट पॉइंट.
One of the DNA strands binds in a cleft in the type 1 topoisomerase, placing it near the active site.
Now see, this is how the type 1 topoisomerase is going to act. Type 1 topoisomerase act like this. Then it moves to the DNA and binds to the active site of the DNA molecule. So you look that the topoisomerase are being called topoisomerase because they have an active site tyrosine molecule present within itself. Topoisomerase का नाम topoisomerase इसीलिए पड़ा है because they contain an active site tyrosine present within their structure. They have a tyrosine molecule, that is why they are called as topoisomerase. Now, what would be the reaction that would be taking place? The tyrosine has an hydroxy group present over itself. The hydroxy group will cause the deprotonation. The other groups will cause the deprotonation of the proton, leading to the formation of an oxyanion. An O negative ion, that is referred to as an oxyanion, will attack the phosphodiester backbone and will cleave the phosphodiester backbone. How? Please look at it.
The active site tyrosine becomes deprotonated, probably by a...
The active site tyrosine has become deprotonated because the proton has been absorbed from the tyrosine OH group, hydroxy group, as the proton has been absorbed from the tyrosine hydroxy group. So there is an alkoxide ion that is produced, that is O negative. This O negative is going to attack the innermost phosphate molecule, that is the alpha phosphate molecule, that will break the phosphodiester backbone. ये अटैक कर जाएगा phosphate molecule के ऊपर और innermost phosphate की जो phosphodiester backbone है वो break कर देगा. Now you might be thinking, sir, which is this basic amino acid residue? But this basic amino acid residue is lysine. You would have known about this particular amino acid residue that is referred to as lysine. Lysine has a structure with epsilon amino group. So you can look at its structure. Within its structure, what you can find is that there is an epsilon amino group. A side chain is present, alpha, beta, gamma, delta, and an epsilon. And epsilon contains an amino group. Okay, it contains an amino group, NH2 group. And it is this amino group that is going to attack. This is alpha, beta, gamma, delta, and this is epsilon naught. And it is this epsilon naught group that will be attacking the other molecules to take place. Okay. It will attack the hydrogen atom of the hydroxyl group present over the tyrosine and will abstract the proton. ये OH ग्रुप के ऊपर अटैक करेगा और एक प्रोटॉन को अपने अंदर ले लेगा. जब एक प्रोटॉन को ले लेगा तो यहाँ पर क्या हो जाएगा बेटा? NH3 positive, and this is the O negative ion that would be produced. यहाँ पर O negative ion produced हो जाएगा. Let us move forward.
A nearby basic amino acid residue. The result is a negatively charged oxyanion,
Okay, so there would be a negatively charged oxyanion. So this is an oxyanion that is going to be produced. Okay, so this oxyanion, once it is going to be produced, then this oxyanion is going to attack the innermost phosphodiester backbone. और ये जब phosphodiester backbone पे अटैक करेगा तो phosphodiester backbone को क्या करेगा, ब्रेक कर देगा. Phosphodiester backbone एक बार हो गया ब्रेक, then what is going to happen, the DNA stability is being lost and the supercoiling present within the structure of DNA is gone. Okay, how it occurs, let's look at it.
The oxyanion attacks the phosphate group at the
See,
3' position
the
of the nearest
oxyanion is going to
deoxyribose,
attack the phosphate
producing
group.
a DNA 3' phosphotyrosyl enzyme intermediate and
So
a
there
5' hydroxyl polynucleotide. The hydrogen.
See this. Once the attack is going to take place, the oxyanion attacks the phosphate group at the 3' position of the nearest deoxyribose. Once the oxyanion is going to attack at the 3' position of the nearest deoxyribose, it will produce a 3' phosphotyrosyl enzyme intermediate. There is a 3' phosphotyrosyl enzyme intermediate that is going to be produced. ये जो इंटरमीडिएट यहाँ पर बन गया है बेटा, इसका नाम क्या है? दिस इज 3' phosphotyrosyl intermediate that has been produced, and this is the intermediate that is going to react further and 5' hydroxyl polynucleotide, and it will form another molecule that is referred to as 5' hydroxyl polynucleotide. तो यहाँ पर ये दूसरा मॉलिक्यूल बनाएगा जिसका नाम है 5' hydroxyl polynucleotide. The hydrogen atom on the 5' hydroxy is probably donated by a nearby acidic amino acid residue. Now see, there is another acidic amino acid residue that will be there. Okay, and this is going to abstract a proton from here. Now what is going to happen? Let's move forward and look at it. अब क्या होने वाला है देखते हैं हम आगे.
Atom on the 5' hydroxyl is probably donated by a nearby acidic amino acid residue.
So there is an acidic amino acid residue, and most probably this acidic amino acid residue is glutamate. So glutamate has an COOH group, that is a carboxylic group, and this carboxylic group has the ability to donate a proton. यहाँ पर एक carboxylic group होता है जो कि ग्लूटामेट का होता है, ये अपना प्रोटॉन डोनेट कर देता है. जैसे ही इसका प्रोटॉन डोनेट होता है, then there is CH2OH group that is going to be produced. Now it is this oxyanion that has been produced which will again recause the formation of the bond once the conformation is changed. एक बार सुपर कॉइलिंग को हटा दिया जाएगा, यही जो एसिडिक ऑक्सीएनाइन प्रोड्यूस हुआ है यही रिएक्शन को रिकैटलाइज करेगा, दोबारा से कराएगा. कैसे कराएगा देखिए सामने.
After cleavage, the enzyme undergoes a large conformational change.
See, after the cleavage, the enzyme undergoes a large conformational change. Okay, so what you find here is that the enzyme is undergoing a large conformational change. इस बात से आपको ये पता चलता है कि क्लीवेज के बाद में enzyme अपने अंदर एक conformational change कर लेता है. Enzyme causes a conformational change in itself. There are two theories given in order to explain the mechanism of enzyme catalyzed reaction. Can you tell me the name of those two theories? One is referred to as the lock and key hypothesis, okay, given by Emil Fischer, and then the Koshland, who gave the next hypothesis, which is referred to as induced fit hypothesis. Now see, according to the lock and key hypothesis, enzyme act as a lock and substrate act as a key. That means there is no three-dimensional conformation changes that can take place in the enzyme. अगर ये enzyme है और ये substrate है तो enzyme एकदम lock की तरह एकदम fix है और fix होके ही substrate से bind करेगा, यानी कि किसी और substrate से bind नहीं कर सकता. As for every lock there is a key, so for every enzyme there is a substrate. But now this hypothesis has been disapproved right from the study of the topoisomerase enzyme. You look here, topoisomerase enzyme undergoes a large conformational change. यानी कि topoisomerase जब bind करता है तो अपने अंदर conformational change लाता है. This is what the other theory that is referred to as the induced fit hypothesis state. Enzyme can undergo conformational change in its active site and can bind to similar molecules. तो यहाँ पर topoisomerase act as a phenomenal proof of the induced fit hypothesis. As topoisomerase undergoes conformational change, as topoisomerase enzyme undergoes conformational change. Enzyme undergoes conformational change. कॉन्फॉर्मेशनल चेंज, जैसे जैसे टोपोमरेज एंजाइम के अंदर कॉन्फॉर्मेशनल चेंज होता चला जाता है, इसीलिए आइसोमरेज एंजाइम इज वन ऑफ द बेस्ट एग्जांपल ऑफ इंड्यूस्ड फिट हाइपोथेसिस. सो इट इज one of the best example of induced fit hypothesis. Okay, please note down this point. Next point that we are going to start. Let us move forward to understand now what further happens.
This conformational change opens up a gap in the cleaved strand, with the enzyme bridging the gap.
Okay, so this is the creation of a particular gap in between the single strands of the DNA. ये एक gap produce कर देता है single strands of the DNA में. Now it is going to cut this, it has already cleaved this DNA. It is going to take these DNA molecule and then place these DNA together with each other and then seal them. उसके बाद में ये दूसरी तरफ ले जाएगा और उसे seal कर देगा. How?
The uncut strand then passes through the gap, binding to a pocket in the middle of the enzyme.
Now,
binding this strand causes the topoisomerase to change conformation again, closing the gap in the nicked strand.
Now, the topoisomerase has again changed its conformation and ultimately sealing the gap. How it is going to seal the gap? जो भी अभी तक रिएक्शन हुआ ही है उसका उल्टा कर देंगे, उसको इनवर्स डायरेक्शन में चला देंगे और गैप को पूरी तरीके से सील कर देंगे. So let us look at it.
The topoisomerase then ligates the nicked strand by...
See, this is what is going to happen. The topoisomerase will now cause the next reaction to proceed. Topoisomerase अब क्या करेगा? अगली रिएक्शन को कराएगा. The acidic group, which was the glutamate acidic group, will abstract a proton from the 5' OH group. As it has abstracted a proton, so what happens? Look here.
By reversing the cleavage reaction.
See this. Once the OH was being removed, एक बार हाइड्रोजन 5' OH से रिमूव हो गया, तो 5' पे क्या बन गया था? 5' पे ऑक्सीएनाइन बन गया था. Okay, as oxyanion was being produced, this oxyanion attacked the phosphodiester backbone again, which was finding its attachment to the active tyrosine residue, and now this phosphodiester backbone has again been produced. यहाँ पर जो टायरोसिन था जिसने उसे ब्रेक किया था उसकी रिएक्शन को उल्टा करा दिया गया है. अब इससे हुआ क्या? जो टेंशन थी ना डीएनए में वो निकल गई है, यानी कि सुपर कॉइलिंग खत्म हो गई है, यानी कि नेगेटिवली सुपर कॉइल्ड डीएनए अब रिलैक्स्ड हो चुका है. नाउ दैट हैज बिकम कंप्लीटली रिलैक्स्ड. It has abstracted its proton and it has remained as such. देखिए ऐसे ही तो था जब ये एंजाइम ने रिएक्शन शुरू की थी और ऐसा ही एंजाइम रिएक्शन के बाद में भी हो गया. This enzyme has this type of a conformation when the reaction had started and it has reached the same conformation when the reaction has finished. So this proves that enzyme remains unchanged during the course of the reaction. Enzyme remains unchanged during the course of the reaction. पूरी रिएक्शन के दौरान एंजाइम हमेशा क्या रहते हैं? एंजाइम always remains unchanged during the entire course of the reaction, whatever the reaction is taking place. चाहे रिएक्शन कैसी भी हो, एंजाइम कभी भी अपने आप को चेंज नहीं करते. एंजाइम पूरे कोर्स ऑफ रिएक्शन में सिमिलरली रहते हैं. दे आर ऑलवेज सेम. ओके, सो लेट्स मूव फॉरवर्ड and this is what is going to happen.
Finally, the topoisomerase undergoes another conformational change, opening up and releasing the DNA.
So now the topoisomerase will undergo another conformational change and it will be releasing the DNA molecule. So this is how topoisomerase works. इस तरीके से type 1 topoisomerase work करता है. Type 1 topoisomerase क्या करता है बेटा, फिर से सोच के बताइए. नेगेटिवली सुपरकॉइलिंग को हटा देता है और towards positive supercoiling move करता है, because as it do not utilizes ATP molecule, so it cannot induce positive supercoiling. At maximum it can make the DNA relax. ठीक है, तो ये क्या कर देता है, DNA which is negatively supercoiled, DNA which is negatively supercoiled will be relaxed. Negatively supercoiled will be made relaxed. Supercoiled will be made relaxed, will be converted into relaxed DNA. ये DNA को relax कर देता है. The negatively supercoiled DNA is getting converted into a relaxed DNA molecule. This is how the interconversion is going to take place. तो इस प्रकार का interconversion यहाँ पर देखा गया है. Okay, so let us move forward.
Overall, the reactions catalyzed by the type 1 topoisomerase have added one twist to the DNA, increasing the linking number by 1.
See, this is what has happened. Previously, the linking number was being defined by the twist, which were referred to as M. But now, after the activity of this particular enzyme, that is topoisomerase type 1, the linking number has been increased by 1. Linking number एक वैल्यू से इंक्रीज हो गया है क्योंकि ट्विस्ट एक वैल्यू से इंक्रीज हो गई.
How well did you learn the mechanisms of the reactions catalyzed by type 1 topoisomerases? In this section, you will find out.
Now, let us solve certain critical question with the type 1 topoisomerase activity. अब हम type 1 topoisomerase की activity के साथ में कुछ interesting questions को देखते हैं, that how well you are able to understand your knowledge with these questions. Okay.
Question 1. What is the first step of the cleavage reaction? Click on the atoms involved in this step.
Now, the first step of the cleavage reaction. You can just memorize that in the first step of the cleavage reaction, सबसे पहले step में क्या होता था? It was a basic amino acid which was lysine, which was causing an attack over the oxygen atom, over the hydroxyl group and abstracting a proton. ये जो अमीनो ग्रुप होता था ये हाइड्रोक्सिल ग्रुप के ऊपर अटैक करता था और प्रोटॉन को एब्स्ट्रैक्ट करा लेता था. Once the proton was being abstracted, then an oxyanion was produced. एक बार ये प्रोटॉन को बेटा ले लेता था तो बेटा क्या बन जाता था? Once proton was being produced, was being taken, then here oxyanion was produced. Oxyanion was produced. There is an oxyanion that is going to be produced. How this oxyanion is going to be produced? Please let us look forward. Yes, it is the tyrosine 1 from where this would be removed.
Correct.
Okay, so this is how the reaction was taking place. You can look forward. See this? This is the reaction. This is an oxyanion production. Now this oxyanion is going to attack the phosphodiester backbone. Okay, moving forward.
Question 2. What is the second step of the cleavage reaction? Click on the atoms involved in this step.
The second step of the cleavage reaction involves the attack of the oxyanion over the innermost phosphate. As the oxyanion is going to attack on the innermost phosphate, then it is going to break the phosphodiester backbone.
Correct.
Okay. Try to answer the questions along with me, as majority of the students are doing. मेरे साथ में questions का आंसर देने की कोशिश किया करिए. आप online classes में तो ऐसा कर ही सकते हैं. Now the next question is here.
Question 3. What is the final step of the cleavage reaction? Click on the atoms involved in this step.
Now, what is the final step of the cleavage reaction? How the final step of the cleavage reaction takes place? क्या होने वाला है final step में cleavage reaction की? Can you tell me what would happen in the final step of the cleavage reaction? Click on the atoms that are going to be involved. Can you just click on those atoms? The final steps of the cleavage reaction involve the oxyanion and the proton from the acidic amino acid. The proton from the acidic amino acid is donated to the 5' oxyanion so that the oxyanion can be stabilized.
Correct.
Moving to the next question. This is how the reaction takes place. The oxyanion has been established and stabilized. Okay. तो इसने प्रोटॉन यहाँ पर दिया था और प्रोटॉन ने इसे स्टेबलाइज कर दिया. Now what happens in the next question?
Question 4. What happens after the topoisomerase cleaves one strand of the target DNA?
Now,
Put these steps in order. You may have to use a step more than once.
first, the what would be the sequence of events that would be taking place? Okay, the uncut DNA strand passes through the gap in the cleavage strand and binds to topoisomerase. The cleaved strand is ligated. See, the cleaved strand is always ligated at the end. Okay, the type 1 topoisomerase undergoes a large conformational changes. Yes, the type 1 topoisomerase undergoes a large conformational change. The uncleaved DNA strand passes through the gap in the cleaved strand and binds to topoisomerase. Okay. The type 1 topoisomerase undergoes a large conformational change always. Okay. There are 4 boxes, so you have to fill the 4 boxes, not one. The cleavage strand is ligated. Let me look here.
Correct.
Okay, so this would be the sequence of event that would be taking place. Again, explaining this particular answer. See, the type 1 topoisomerase to be cleaved, the following reaction has to take place. Just wait for a second. This is what is going to happen. The type 1 topoisomerase undergoes a large conformational change, cleaving the single-stranded DNA molecule. The single-stranded DNA molecule is made to pass through the second strand just adjacent to it. पहले तो सिंगल स्ट्रैंडेड डीएनए मॉलिक्यूल को कट किया जाएगा, उसे सामने वाले स्ट्रैंड से पास कर दिया जाएगा, फिर से कॉन्फर्मेशनल चेंज होगा टोपोआइसोमरेज में, उसे सील कर देगा एंड देन द टोपोआइसोमरेज एक्टिविटी विल बी लाइगेटेड. See the second step, the third step, the fourth step, and this is how the ट्विस्ट नंबर इज गोइंग टू बी इंक्रीज. इस प्रकार से ट्विस्ट नंबर इंक्रीज हो जाएगा. तो इस तरीके से टोपोआइसोमरेज ने यहां पर आपके लिए ट्विस्ट नंबर को इंक्रीज कर दिया है. दिस इज हाउ इट अकर्स. नाउ टू द नेक्स्ट क्वेश्चन.
Question 5. What is the first step of the ligation reaction? Click on the atoms involved in this step.
Now, how the ligation reaction is going to take place? What will happen in the case of the ligation reaction? For the ligation reactions to take place, what is going to happen? This hydroxy ion and this the carboxyl group ion are going to react together with each other. When the proton will be abstracted by the acidic amino acid residue, then the alkoxide ion produced will attack the innermost phosphate, and this will create the reaction.
Correct.
This is how the abstraction of proton takes place. Oxyanion produced. Oxyanion will make the reaction to take place.
Question 6. What is the second step of the ligation reaction? Click on the atoms involved in this step.
It's going to attack the innermost phosphate molecule. Okay?
Correct.
Yes, you are also correct, Shivangi. Good answers.
Question 7. What is the final step of the ligation reaction? Click on the atoms involved in this step.
In the final step, the oxyanion will abstract its proton from the basic amino acid residue.
Correct.
It has abstracted its proton.
Question 8. A negatively supercoiled DNA molecule has a linking number of 55. What is the linking number of this molecule after one cycle of relaxation by a type 1 topoisomerase?
After one cycle of relaxation by a type 1 topoisomerase, the linking number is going to be changed to 56 because it is going to increase the linking number. So the linking number is always going to be increased by one value. These types of questions are asked in GATE entrance examination very often. Okay.
Correct. The topological state of DNA.
Now we have reached towards the conclusion of the type 1 topoisomerase. Now we would be ending the type 1 topoisomerase exercise. Please look at the conclusion that how does the topoisomerase works. A very simple exercise was this. So please do pay your attention here.
Is controlled by a class of enzymes called topoisomerases. Type 1 topoisomerases relax supercoiled DNA by increasing the linking number one step at a time.
Okay, so this type 1 topoisomerase is going to act one step at a time. One step it is going to work and then another step and so on. Next point.
In order to increase the linking number of a DNA molecule, many type 1 topoisomerases must first bind to a segment of DNA in which the two strands are melted. The topoisomerase then cleaves one of the two strands.
Topoisomerase then cleaves one of the two strands, changes its conformation and reseals the gap.
Next, the type 1 topoisomerase passes the other strand through the nick and ligates the nicked strand.
Yes, this is how nicked strands are being ligated. ये जाता है, nick करता है और फिर ligation करा देता है. And so this is how ligation is being achieved.
Overall, the reactions catalyzed by the type 1 topoisomerase add one twist to the DNA, increasing the linking number by 1.
So they are going to increase the linking number by a value of 1. ये linking number को एक value से बढ़ा देते हैं. They are going to increase the linking number by a value that is equivalent to 1. Next.
You have completed this exercise.
This is the ribbon diagram of the type 1 topoisomerase, that how it acts. It is the one that is melting the DNA molecule and causing the one strand to pass over the another strand, increasing the linking number by 1. Type 1 topoisomerase is increasing linking number by 1. Type 1 topoisomerase cleaves one strand of the melted DNA. Next, the type 1 topoisomerase passes the other strand through the nick. Type 1 topoisomerase increases twist by 1 and therefore increases in linking number by 1. And the most important point about the type 1 topoisomerase is that they remove negative supercoiling. They removes the negative supercoiling. Removes the negative supercoiling. Negative supercoiling by introducing positively supercoiling. By introducing positive supercoiling. By introducing an increase in linking number. But this is very important. These topoisomerases, but type 1 topoisomerases can only relax negatively supercoiled DNA, but cannot induce positive supercoiling. ये कभी भी positive supercoiling को induce नहीं कर सकते. Cannot induce positive supercoiling within the DNA structure. Okay, so they are never going to introduce a positive supercoiling within the DNA ultrastructure. This is how the topoisomerases, especially the type 1 topoisomerases, are going to act. This is all about type 1 topoisomerases. We have concluded the end of this type 1 topoisomerases, end of today's lecture. Thank you. Thanks a lot. Good night. Bye bye. If you have any question, you can always write it in the chat box.